All right, and now let's go to the next section of the paper results. This is on detection and isolation of a new, quote, virus. So now let's take a look at the next part of the paper results, this time on supposedly, quote, detecting and isolating a new virus. And here it is, the detection and isolation of a novel coronavirus, and these are the results of these detections and isolations, supposedly. So 3 bronchoalveolar lavage samples were collected from Wuhan Jingtan Hospital on December 30th, 2019, and no specific pathogens, including this is a human coronavirus 229E, HCoV, human coronavirus NL63, then this 0C43, and this other one, HKU1, were detected in clinical specimens from these patients by the RespiFinder Smart 22 kit. And then RNA extracted from bronchoalveolar lavage fluid from the patients was used as a template to clone and sequence a genome using a combination of Illumina sequencing and nanopore sequencing, and more than 20,000 viral reads from individual specimens were obtained, and most contigs matched to the genome from lineage B of the genus Beta coronavirus, showing more than 85% identity with a bat SARS-like CoV, and its name is here bat SL, and then CoV ZC45, and then MG772933.1, genome published previously. So yes, they basically found some pneumonia patients, took some airway fluid samples, and then used some genetic testing kits to detect if there's these previous supposed coronavirus, human coronaviruses. I believe these are the common flu ones, or the common cold symptoms, and so on. But then no specific pathogens were found, supposedly, and then instead they took RNA from that fluid and then used it as a template to clone and sequence a genome using these 2 sequencing technologies. And they got 20,000 reads, and then most of the contigs or the continuous sequences from those. So you have all these all of the sequences, and then you have then you'll make a continuous sequence from this right here. Then all these overlapping ones, you'll make it, and and then you form that into one giant sequence. It's a contig, or that's contiguous. So match the genome from lineage B, and yeah, that shows 85% identity with this bat SARS-like CoV. All right, and again, here these types of coronaviruses are types of human coronaviruses, and going of known human coronaviruses. And also this right here, this RNA template to clone, uses a template to clone, refers to creating DNA from a specific RNA sequence, and then amplifying the DNA to make amplifying DNA to make many copies. Yeah, so you would use the RNA as a template, produce the complementary DNA, and then amplify DNA to make many copies. And then viral reads, the 20,000 viral reads, are short DNA or RNA sequences generated after breaking down, quote, viral genetic material into small fragments, and then using high throughput sequencing technology to sequence the fragment. So yes, a whole lot of sequencing going on. All right, continuing further into the paper. So positive results were also obtained with use of a real-time reverse transcription PCR assay, or RT-PCR assay, for RNA targeting to a consensus RD-RP region of pan beta coronavirus, or beta CoV, although the cycle threshold value was higher than 34 for detected samples, and assuming these are high threshold values there. And virus isolation from the clinical specimens was performed with human airway epithelial cells and VERO, E6, and this HA7 cell lines. So, so these supposed isolation techniques were done not just on the human airway epithelial, but also on these VERO and these HA7 cell lines. And the isolated virus was named 2019 nCoV. All right, and now this PCR cycle threshold. So this one here, for this test, they used a relatively high number, even as stated from them right here, although the cycle threshold value was higher than 34 for detected samples. So the PCR cycle threshold, or CT value for short, is a specified maximum number of PCR cycles allowed for a positive reading to be accepted when detecting a specific DNA or RNA sequence. And it can also be used to quantify the amount of DNA or RNA in a sample. And here is a chart of that. So here's the source from this random website. So illustration of cycle threshold CT for, or C and then lower t like that. Yes, capital C, lower t, for specifying a sample as positive or negative for a targeted sequence. So in the PCR one, if you're amplifying, and then each cycle you're going to be amplifying a lot of the DNA that you're replicating. So you're going to replicate, it's going to get a whole bunch, and then after a set amount of cycles, the CT value, and also there's a fluorescence threshold right here. So the way it works is once you amplify enough, and then if it gives off a specific fluorescent radiation emission, and then it'll be considered as positive. But then the number of cycles to get to that point, if it's above the CT value, then you're not going to have a positive sample. Yeah, for example, here, so the CT is right here, and then this sample right here, it was able to cross this threshold, or this fluorescent threshold, before or right at the CT maximum cycle threshold, and then now this is considered positive. But then these other ones, this other one that you're amplifying a whole bunch of times, but then it never reaches this threshold, is a negative sample. And this other one that goes above this, but it took you way too many cycles, and this one is 35.19 PCR cycles. So there's a fraction there. So this is still considered a negative sample. So this one you only need to amplify a tiny bit. So this just means there's more of that sample inside this one to get this positive reading, like that. So, and interestingly, this example had 35.9 as a negative sample. This one is a 34 still. So, so then I would assume it's going to be on the high end as well. All right, going further. So right here, so VERO E6. So VERO E6. The quote virus isolation was performed on the human airway epithelial and VERO E6 and HA7 cell lines. So VERO E6 are a type of cell line derived from the kidney cells of an African green monkey in 1968 as the VERO 7-6 cell line, and then cloned in 1979 to form the VERO E6 cell line. And these cell lines are commonly used in quote virus isolation because they are highly permissive to quote viral infection. And a cell line is a population of cells that are derived from a single cell and can be maintained and grown indefinitely in culture. So it's a population derived from a single one, and you can keep maintaining, growing them, and so on. And HA7 is a cell line derived from a liver cancer. From the liver cancer, or fix that up, from liver cancer cells of a 57-year-old Japanese male in 1982. All right, that's interesting. And then cloning cell lines is the process of creating genetically identical cells from a single parent. All right, it's very, very interesting. So basically, they would do this quote virus isolation experiments or procedures on the human airway epithelial cells of the patients. These are of the cultures, as well as these green monkey cells, or these cell lines are just these culture cells, from kidney cells of an African monkey, and then that was cloned and kept going for many years. And also this HA7, this is a liver cancer patient, a Japanese 57-year-old Japanese male with liver cancer. And then he took cells from his liver cancer, and they grew that in a culture, and you get a cell line of those identical cells. Interesting. So continuing further in the paper, to determine whether virus particles could be visualized in this 2019 HCoV infected airway epithelial cells, mock infected and 2019 HCoV infected human airway epithelial cultures were examined with light microscopy daily and with transmission electron microscopy 6 days after inoculation. So this mock infected, they didn't actually explain how they did that. They just stated that. So cytopathic effects were observed 96 hours or 4 days after inoculation on surface layers of human airway epithelial cells. and there's a lack of cilium beading was seen with light microscopy. Yeah, I believe they just spelled this wrong. There should be an S somewhere there, but this is, I just copied it from the paper. So seen with light microscopy in the center of the focus, figure 2, and no specific cytopathic effects were observed in the VERO E cell and Ha7 cell lines until 7 days after inoculation. So here's figure 2, cytopathic effects in human airway epithelial cells after inoculation with 2019 HCoVs. So this one's mock infected. They don't explain how. They just do that, and then there's just a bunch of uniform dots there. And now the center of this focus, this is the human airway epithelial cytopathic effects, and it just has this this light contrast area that's supposedly the cilium beading. And mock infected is a control group in which the same procedure is done but without the actual virus. Well, that's the definition, but I don't know what they actually did. And then cilium beading refers to the coordinated movement of cilia, or plural of cilium. And remember, these are the ones that move the mucus and particles out of the respiratory tract. And then this one is they're not moving in coordinated form anymore. But again, this is just a blurry image of something. So continuing further in the paper, electron micrographs of negative stain 2019 HCoV particles were generally spherical with some pleomorphism, so it's having different shapes. Figure 3. So diameter varied from about 60 to 140 nanometers, and virus particles had quite distinctive spikes, about 9 to 12 nanometers, and gave virons the appearance of a solar corona. So these apparently this is the virus of virons, and then there's a solar corona around somehow. All right. Extracellular free virus particles and inclusion bodies filled with virus particles and membrane-bound vesicles in cytoplasm were found in the human airway epithelial ultra-thin sections. This observed morphology is consistent with the Coronaviridae family. Alright, so now we have this figure right here, and this is the main, quote, evidence, the physical evidence, besides the sequencing, that they're showing that there's a supposed virus. So, visualization of this, 2019 HCoV with transmission electron microscopy. So you have negative stained 2019 HCoV particles. This is from the supernatant, is in panel A. Yeah, are shown in panel A. So panel A is a supernatant, and you have these right there. Yeah, those are the supposed virons and the supposed solar corona around it. And you also have on the part B, this is the ultra thin sections of that in that resin block that were cut. And 2019 HCoV particles in the human airway epithelial cell ultra thin sections are shown in panel B. Arrowheads indicate extracellular virus. So those are the arrowheads. No, the arrowheads is right here. So these ones are assuming that these are got out of the cells. And this is another one, arrow. Supposedly these little dots are viruses. All right. And what else? Arrows indicate inclusion bodies formed by virus components. So there's inclusion bodies or supposed virus factories inside the cells that somehow produce these supposed viruses. And going further, and triangles indicate cilia. So these are the cilium beading here. All right, this is black and white. Remember, these are all dead images and then stained as well. All right. All right, continuing further. So pleomorphism, it's over here. So some of these show pleomorphism, just showing different shapes. Refer to the variability in the size and shape of cells or their nuclei. And then this solar corona, so the solar appearance of a solar corona, or solar or stellar corona refers to the outermost layer of a star's atmosphere. And during a total solar eclipse, it can be seen by the naked eye. And a total solar eclipse occurs when the moon completely blocks the sun from being seen from a point on the Earth. And extracellular free virus particles are quote, viruses found outside of and released by, quote, infected cells. And these supposedly can be spread through coughing, talking, sneezing, and inhaled by others to start a new, quote, infection. All right, virus inclusion bodies or viroplasm are supposedly, quote, viral factories where the, quote, virus replicates and assembles. So these inclusion bodies are supposedly where these viruses are being produced, supposedly. All right, membrane-bound vesicles are structures within cells that are enclosed by membrane and have various purposes, such as transporting, storing, or breaking down material. And the cytoplasm is all the material within a cell. So membrane-bound vesicle, that was over here. So those ones, the virus particles, are in the inclusion bodies in membrane-bound vesicles. And going further, here is a solar corona. Yes, the solar corona there. I didn't put any notes there. So this is the solar corona around the. This is when the moon blocks the sun, and now you have these rays. All of these stuff around it, the corona, around the sun there, that's visible in the naked eye, as stated here. Yeah, so a total solar eclipse occurs when the moon completely blocks the sun from being seen from a point on the Earth. And this is just right to here. Total solar eclipse. And here I just wrote that down. So total solar eclipse, making the solar corona visible with the naked eye. And here's another illustration of a solar eclipse. So you have the sun here. And again, this is from a point on the Earth, that's how it will be visible. And then you have the shadow of it. The shadow, the inner one, is called the umbra. But then whereas the outer one here, cutting from this angle, you have this penumbra. There's an orbit of the Earth, and then orbit of the moon around like that, and you're going to have eclipses and so on. And here I just wrote that down. So illustration of a solar eclipse. All right, then now let's continue further and now look at a summary of this, quote, new virus detection and isolation from the paper results. So here's a summary of the paper results on detecting and isolating this quote, new virus. So 3 airway fluid samples were collected from a Wuhan hospital on December 30th, 2019. And no specific, quote, viruses were detected from these patients or specific known viruses were detected from these patients. And an RNA was extracted from the airway fluid samples and assumed to be from a, quote, virus. The RNA was used as a template to create DNA, which was later amplified to many copies, and then sequencing technology was used to produce a genome. All right, and then 20,000 short RNA sequences were obtained, and long continuous sequences from these short and overlapping sequences, i.e. contigs, were produced and showed 85% similarity with a bat coronavirus. And then PCR tests using a cycle threshold CT value higher than 34 were positive when using a known sequence common among beta coronaviruses. So first of all, they did tests and they showed that no specific known viruses were detected from those patients. And then they showed sequences from beta coronaviruses that were shown inside samples using these PCR tests. And then the overall long continuous contigs, or pretty much genomes, showed 85% similarity with a bat coronavirus. And, quote, virus isolation from the clinical sample specimens were performed on human airway epithelial cells, monkey kidney cells, and liver cancer cells from a 57-year-old Japanese man. Cancer. So yeah, monkey cells and also from liver cells from a 57-year-old man. And the, quote, isolated virus was named 2019 HCoV, or 2019 novel coronavirus. And the, quote, virus was then used to, quote, infect human airway epithelial cell cultures with a, quote, mock infected culture used as a control, although there's no information shown in the paper about this. They just state it, but they don't actually show anything else. The cultures were examined daily with optical light microscopy and with TEM, or transmission electron microscopy, 6 days after, quote, infection. And cytopathic effects were observed 96 hours, or 4 days after inoculation. And also, I just want to go back here. So the cultures were examined daily with optical light microscopy and with transmission electron microscope, 6 days. I think this means 6 days after infection, or 6 days. So you would basically, because it's doing it daily, I think there's the word 4, but then if you scroll back up to what it actually said, they never actually said here. So with transmission electron microscope, 6 days after inoculation, but it's searched daily. And I'm just going to go 4 ? I'll just put that in. I think that's what it means. Because the cytopathic effects were observed 96 hours after inoculation. even though this says 6 days after. So obviously it was doing it daily. All right. Yeah, so we corrected that part of the paper. So, yeah, cytopathic effects were observed 96 hours or 4 days after inoculation on surface layers of human airway epithelial cells, and with a lack of coordinated cilium movement was observed in some spots, that's cilium beating. In the monkey kidney and human liver's cancer cell culture, cytopathic effects were observed 6 days after COVID infection, as opposed to 4 days for the human airway samples from the patient. So, and here's the images. There's the mock. There's the human airway epithelial showing the cytopathic effects. Again, it just shows just a little low contrast area. So figure 2, supposed cytopathic effects, CPE, in human airway cell culture on the right image with a lack of cilium beating in the center. And there's the mock on the left. For the supernatant samples, electron microscope images of, quote, virus particles right here were presented, in which the background was stained with heavy metals. The particles varied in size and shape and ranged between 60 to 140 nanometers, supposedly with spikes 9 to 12 nanometers in size, and supposedly looking like a solar, quote, solar corona. This just looks like just something. For the embedded and heavy metal stained / fixed thin sections of human airway epithelial cell cultures, electron microscope images showed quote, viruses. I'll put this right here. Yeah, put, quote, viruses outside the cells, as well as within membrane-bound structures inside the cells that supposedly act as, quote, virus factories in the replication and formation of, quote, viruses. The observed, quote, virus particles supposedly belong to the Coronaviridae family. So, right, so here's the 2 images. So negative stained supernatant on the left, supposedly showing, quote, virus particles. And on the right shows the ultra-thin embedded sections, inclusion bodies or, quote, virus factories within cells. That's the arrows. There's extracellular viruses, arrowheads, and there's cilia, the triangles right here. All right. So yes, so this is the supposed evidence of a new virus that's supposedly infecting the world. And to me, this is not evidence.